like to thank Dr. Tonu and his team for inviting me to talk for livestock, because I realized all the preceding presentation was on human health. And I'm going to give a twist. I'll be talking about gene editing for enhancing animal health, productivity, and resilience. So the first segment of this is on vaccine development for African swine fever. So this disease is caused by a virus, which is a lethally hemorrhagic fever disease in pigs, which causes up to 100% mortality, depending on the genotype of the virus. Currently, there are no vaccines, and there are also no effective treatment against this disease. Containment measures that is in use is mostly on slaughter and disposal of infected or potentially exposed animals. And therefore, the approach for establishing these biosecurity measures in sub-Saharan Africa is not feasible because of the cost implication. And mostly the pig farmers in our part of the world is smallholder pig farmers, and they therefore lack the resources to invest in biosecurity. There has been recent outbreak of this disease elsewhere on the globe, and this has really, the estimate is it has taken a quarter of the world's pig population off the market, which has led to socioeconomic issues. Closer home in Africa, this disease is present in 26 African countries, where there are all 24 genotypes. The challenge with that is there could be lack of protection between genotypes. You could generate a vaccine for one genotype and it may not protect other genotypes. Of course, again, there's negative consequences significant in smallholder setting. So there has been approaches to developing vaccines for ASF. One is the conventional or pre-gene editing technologies. The commonly used is what we do is serial passage of the virus in tissue culture. So you keep growing the virus in tissue culture until it loses part of its genome, it becomes weak, and then you can use that as a vaccine. The other approach is what we call recombinant live vaccines. You target genetically, and after identifying some virulent genes, you remove them, and then you end up with a weakened virus that you can use as a vaccine. However, these two approaches have huge bottlenecks. There could be issues of unpredictable or undesired outcome, which is, for example, the deletion or derangement of the genome, which cannot be predicted beforehand. And then that will also lead to loss of virulence and loss of protection capacity. And these approaches are usually very, very slow. It will take up to a year or even more to generate a single vaccine candidate. Therefore, we at ILRI, we employed two approaches. The first one is what we call synthetic biology static genomics, whereby you know the genome sequence of the virus, then you chop into pieces, and then you reassemble them, and you remove the virulent segment of the genome. And that you do through yeast and E. coli, and then you reassemble the virus and make it infectious. And thereby you end up with a synthetic virus that can be used as vaccine. I'll spare you the technicalities of this because of the mixed audience. The second approach is CRISPR-Cas gene editing, whereby you target specifically the gene that you want to remove, and then you remove that in a targeted manner. And we've shown this. We've shown this through this publication, I think in 2020. And through this approach, we've generated, I think, 12 vaccine candidates now. And among the ones that we've tested, we have 2 promising candidates which have 100% safety and efficacy. So, and this is ready to proceed into next level for regulation and even testing cross-protection between different genotypes that is present in Africa. Besides, based on the success that we've had with ASF, we also expand this technology to East Coast fever. These are parasitic disease in cattle, and The socioeconomic impact of this is estimated to be 0.5 billion US dollars as at last year September estimates. And besides these consequences, animals that recover from ECF have low productivity in terms of weight loss, less milk production, less growth power, reduced fertility, and delays in even reaching maturity. The current vaccines that is being used is what we call infection and treatment method, and this is usually co-administered with antibiotics. Therefore, that's a limitation because there's the potential emergence of antimicrobial resistance, which is also a global issue that we're trying to fight. The second challenge with this approach is the difficulty to administer these vaccines in the field. So at ILRI, we are now employing CRISPR-Cas technology to generate a rationally attenuated vaccine so that we can be able to circumvent the need for antibiotics. Right. Besides vaccine development, we are also looking at other important issues in animal production. And what we are trying to do is enhancing elite traits for improved productivity and resilience. And this is a collaboration between ILRI and the University of Edinburgh through a platform called Center for Tropical Livestock Genetics and Health, whereby we are looking at, genome editing assisted precision breeding to offer opportunity to generate livestock that have improved productivity, for example, milk, egg, that can be resistant to diseases or tolerant to diseases, or intensification of local breeds that have elite traits, or generation of livestock that can withstand harsh climatic conditions, that is mostly in Africa, and also mitigating the environmental impact of livestock farming by modifying the host microbiome, the gut microbiome, so that we can be able to reduce methane emission. Right. Besides the science that we do in the lab, we also trying our best to influence policy at continental level. In April this year, myself and another team from Africa, and even some from Europe, we published a paper in Nature Biotechnology, which is talking about genome editing, making genome editing a success story in Africa. And on this, we are addressing the issue of potential of genome editing innovation in Africa and how to overcome roadblocks for genome editing-based innovations in Africa. And this one will be followed by a policy brief that we are now actually working on, that we are addressing to Pan-African Parliament on how to harness the value of genome editing for Africa. Thank you.